Hello, everyone. Um, uh, Welcome to the online session for advances in cell biology. We have previously talked about the synthesis of secretory proteins and about the co-translational translocation across the ER membrane, where we discussed that the signal recognition particle would recognize a signal sequence and recruit the entire complex, including the ribosome, the mRNA molecule, and this particle itself onto a translocon. It will associate with the translocon and will start pushing the polypeptide chain into the ER lumen. And then later a signal peptidase would cleave the signal sequence and rest of the protein is translocated into the lumen of the ER. And this folded protein would then be targeted to its destination via Golgi apparatus. So proteins would move from the ER towards the Golgi apparatus and from the Golgi apparatus towards their destination. We have also discussed that not all the proteins are like soluble proteins. Some of the proteins also tend to stay inside the plasma membrane, be it membrane of ER, membrane of the entire cell, the plasma membrane, or its membrane of mitochondria, the membrane of lysosomes. So all of these membranous structures tend to contain some membrane associated proteins. Sometimes these are peripheral proteins, either associated to the inside of the plasma membrane or to the outside of the plasma membrane protein, or these are transmembrane proteins. Means these proteins are passed through the membrane all the way through the membrane. So they got three domains: a domain that lies outside the membrane, the domain that lies towards the inside into the cytosol, and the part of the protein that stays embedded into the lipid bilayer itself. So these proteins, which become part of any of these membranes, be it a membrane of ER itself, the membrane of Golgi apparatus, the membrane of the mitochondrion, the membrane of the nucleus, or the membrane of the cytoplasm itself. These proteins are recruited onto these membranes at the time of synthesis, and we have discussed this previously, that these could be single pass membrane proteins which pass through the membrane one time, or these double pass membrane proteins which pass through this membrane two times, and then multi-pass membrane proteins. And we have discussed this as well, how this transfer in case of single pass transmembrane proteins, double pass transmembrane proteins, or even multi-pass transmembrane protein takes place. We have discussed all these things in our previous lectures that all of you have attended physically. I think we ended at these two aspects of protein translocation. One was co-translational translocation, where translocation of the protein takes place at the time of translation. So as soon as the polypeptide chain is synthesized, it keeps on growing into the lumen of the ER. And then we talked about post-translational translocation, which often takes place in the mitochondrion, chloroplast, nuclei, and peroxisomes. Means these proteins would be synthesized by the free ribosomes into the cytosol, and then later translocated into any of these organelles, like mitochondrion, chloroplast, nuclei, and peroxisomes, while co-translational translocation often takes place inside the ER. So when they need to be transported later into any of these organelles, what we call post-translational translocation, then proteins are synthesized on the free ribosomes and then later translocated onto these translocons. And these translocons now exist not on the ER but in the membranes of mitochondria, chloroplast, nuclei, and peroxisomes, etc. So again, what we need is a translocon for the recruitment of these proteins into any of these organelles. We previously discussed that these proteins, which are synthesized by the free ribosomes in the cell, could be needed or they're generally needed by the cell itself, but they could be needed at different locations, like inside the mitochondria, Golgi apparatus, ER. Some of them stay inside the cytoplasm, some of them are recruited to the nucleus. But for recruitment of these proteins, we need to these destinations, we need structures, what we call translocons. So these structures are generally composed of different proteins, and these are complexes. It's not just a single protein, but generally most of the times these are complexes. So there are three different ways in which proteins can be translocated through different types of translocators. One type of translocator we have discussed in details, functions for generally for the ER, and this is known as Sec61 complex, this translocon. So this is a complex, this is Sec61 protein. Sec perhaps stands for secretion or secretase, etc. I'm not sure about it, but that's Sec61 complex. So this is a translocon. And Sec61 protein exists in almost all forms of life. It exists in bacteria, in archaea, and in eukaryotes. So both in prokaryotes and in eukaryotes, you can find this type of translocon, that's Sec61 complex. We do not have ER inside bacteria or archaea inside in prokaryotes. We do not have these membranous structures, but we do have this protein, this Sec61 complex. And we also have the signal recognition particle and signal recognition receptor. So all these three structures, the Sec61 complex, the translocon, this type of translocon, the signal recognition particle, and its receptor. So these three things are found in all forms of life, and they're part of this co-translational translocation. And we have post-translational translocation. So I think you're pretty familiar with the co-translational translocation. We have previously discussed this in quite some details. One thing I would like to add that in case of co-translational translocation, no energy is needed to push the polypeptide chain into the lumen. While in case of post-translational translocation, in both of these two cases, one system that exists in the eukaryotes and the other one that exists in prokaryotes, like in bacteria, both of these two mechanisms, they involve the utilization of some energy which is provided by the ATPs. So while in case of co-translational translocation, no energy is needed, because the only passage that this polypeptide can get through is this translocon, and there's no other way out. So the addition of the new amino acid molecules at the C-terminal basically pushes the N-terminal into the polypeptide, into the lumen, pushes the polypeptide chain into the lumen. So that's the only way, this is the only passage that the polypeptide chain will grow through, and it would directly move into the lumen of the ER. While in case of post-translocational translocation, as we have discussed, that you do not see any ribosomes associated with these two complexes, and the reason why we don't see ribosomes associated with them is because these are the polypeptide chains which have been synthesized somewhere in the cytoplasm by the free ribosomes, and then later recruited onto these translocons. Let's look at this translocon, which exists in the eukaryotes only. And this type of mechanism generally takes place inside the mitochondrion, when you need to import a protein which was synthesized inside the cytoplasm and then later recruited into the mitochondrion, generally passes through the translocon in this fashion. And here we have got, next to Sec61 complex, we have got another complex where four different types of proteins are involved, Sec62, 63, 71, and 72. So it's an additional complex which helps in the recruitment of a pre-synthesized polypeptide chain into the lumen of the mitochondrion, for example. Could also exist in some other cellular organelles, but this is just one example. So what this complex is going to do, Sec62, 63, 71, and 72 complex, this complex recruits binding proteins and drops them onto this growing polypeptide chain. Now these binding proteins have the ability to bind to the polypeptide chain and then release from it in an ATP dependent manner. So hydrolysis of ATP molecule releases a binding protein, and then binding protein needs the ATP molecule to bind to the polypeptide chain and then pushes. So basically these binding proteins, they keep rotating in this fashion. They bind to the polypeptide chain and then they dissociate from the polypeptide chain. They would bind again and then dissociate. So they will keep moving in a wheel-like fashion. And this movement, the binding and dissociation, this keeps pulling this polypeptide chain into the lumen of the mitochondrion or any other organelle in such a way like you would push a rope through a hole in the roof. So if a rope is hanging through a hole into a room and you're standing here, so you got two hands, you grab with one hand, you grab this rope with one hand and then pull it downwards, and then you move your other hand to the top, and then you again with your left hand, let's say you grab the rope and then you pull it downwards once again, and then you grab the upper end with your right hand. So you keep shifting with your right and left hands, and then you keep pushing this you keep pulling this polypeptide chain into the lumen. So that's how these binding proteins keep pulling this polypeptide chain into the lumen. Another type of translocator that we have, so both of these two translocators, they work in post-translational translocation of the proteins. Means pre-synthesized polypeptide chains are pushed through this mechanism into the organelles or to the destination where they're needed. So in this case, this type of translocator exists only in prokaryotes, and this type of translocator exists only in eukaryotes, while the Sec61 complex, which exists in case of co-translational translocation, this exists in all forms of life. So what exists in bacteria? So we know that prokaryotes do not have any structures, membrane-bound structures, but in this case, this would push the polypeptide chain out of the bacteria. So that's why you see it's written cytosol, and rather than any lumen, it's written extracellular space. It means a protein that has been synthesized inside the bacterium is being pushed out of the bacterium into the surrounding medium. In this case, another complex known as Sec-A ATPase, that's the complete name, Sec-A ATPase. ATPase name comes from the hydrolysis of, because this enzyme is dependent on ATP hydrolysis, so it brings about ATP hydrolysis, and this hydrolysis of ATP molecules leads to the up and down movement, a piston-like motion, like you have this piston inside a car's engine, which keeps moving up and down and runs the engine. So the same way, this piston keeps moving up and down with the hydrolysis, each time with the hydrolysis of an ATP molecule, and it keeps pushing this polypeptide chain into the lumen of the out of these bacteria. But you would also find this inside the thylakoid membranes in chloroplasts, because we have previously discussed that chloroplasts and mitochondria, you see these two different mechanisms. So because we have previously discussed that the mitochondria or the chloroplasts are basically result of evolutionary mechanisms where independent bacteria or independent prokaryotic cells were basically eaten up by another cell, and they started living together later and became part of the bigger cell. One turned into a mitochondrion and the other one turned into chloroplasts, and chloroplasts also have these membrane sac-like structures, what we call thylakoids, and you can also find this type of mechanism on the in the membranes of the thylakoids, and this is the reason. This is another reason why or I don't know. I mean, evidence you can call it of the evolution of these chloroplasts and mitochondria, that they were basically prokaryotes, because what exists on the surface of bacteria also exists on the surface of chloroplasts. So this piston-like motion of sec AATPase pushes the polypeptide chain into the lumen, out of this into the extracellular space in case of bacteria, and into the lumen of the thylakoids. And with each push, this enzyme is roughly pushing 20 amino acids into the lumen of the chloroplast, or into the extracellular space in case of bacteria. So these were three ways in which proteins could be translocated through the translocators. In case all forms of life, for co-translational translocation, Sec61 complex, this protein exists in, this is Sec61. So this protein exists, Sec61 exists in all forms of life, while 62, 63, 71, 72 complex exists in eukaryotes only, while Sec AATP as mechanism exists in prokaryotes only. And the reason why it exists in plant cells is because of the origin of the chloroplast molecules. So that's an exclusion. I have added some text, you can read it yourself. We have discussed that these proteins are now being, let's go back to the ER, that these proteins are being synthesized and they're being folded inside the ER. This folding is necessary so that proteins can perform their normal functions. So if they're not properly folded, these proteins may not be able to perform their normal functions. And sometimes what happens is that these proteins are not properly folded, or they remain unfolded. When they're unfolded, some other types of proteins, what we call chaperones, these proteins, you might be familiar with them. These are the proteins which are involved in folding, unfolding, assembling and unassembling of different other macromolecular structures. So these small proteins, what we call chaperones, would bind to these unfolded proteins and will fold them into their proper secondary or tertiary structures. One such example of first type of chaperones, I would say, would be the folding of histone proteins when nucleosomes are synthesized and DNA is wrapped around them. So that folding, that's the place where first of all these chaperones would play a role. But inside the and even inside the cytosol or inside the lumen of the ER, these proteins are needed so they can properly fold the proteins into a structure that is desired for those proteins. But sometimes these proteins could also be misfolded. So what happens with, so the proteins which are properly folded are targeted to their destination, but sometimes they are misfolded. And if they're most misfolded, what happens with the proteins which get misfolded is here. So this is a very interesting phenomenon. The misfolded proteins basically they activate a transmembrane kinase. So this is a transmembrane protein, and it has got the domain of the protein which is towards the cytoplasm. So this domain of this protein, let's discuss here. So this domain which is towards the inside of the ER or into the lumen of the ER, that works as a ligand, and the domain that is towards the cytoplasm basically has got this endonuclease or ribonuclease activity, basically. We have previously discussed that these transmembrane proteins can have enzymatic functions. We have also talked about some functions previously, like like here, that this transmembrane protein has got a peptidase activity, means it can cleave the peptide, a polypeptide chain, and the same way we have talked about ATP synthase, which exists inside the in the membranes of the mitochondrion, and it is involved in ATP synthesis. And here we have an example of transmembrane protein kinase. So as the name indicates, it has got some phosphorylation activity. It is capable of phosphorylating itself. So generally these type of proteins which have this kinase activity, they tend to exist as dimers. So this is a monomer, and there would be another monomer, but they would stay apart from each other at some distance, and binding of the signal basically leads to the dimerization of the two monomers. So this is one monomer, and here would be another monomer, and if these proteins are activated, and in this case they are activated by the binding of the misfolded proteins, so these proteins tend to activate these protein kinase molecules, these monomers. They bind with each other, and this leads to the dimerization of these two proteins, and they phosphorylate each other, and then they generate a ribonuclease activity. When they generate a ribonuclease activity or an endoribonuclease activity, the cytoplasm contains some pre-mRNA molecules, and these are the pre-mRNA molecules for chaperones, which exist inside the cytoplasm. So what happens, that this ribonuclease domain of these transmembrane proteins, of this dimer, this ribonuclease domain would remove the intron from this pre-mRNA molecule. So intron is removed, and the two exons, or the two ends, they are joined together to constitute a mature mRNA molecule where intron has been removed. Once this mRNA molecule is synthesized, which is an mRNA molecule for a regulatory protein, a gene regulatory protein. I'm sorry, this is not a pre-mRNA molecule for the chaperones. Rather, it's a pre-mRNA molecule for a regulatory protein. And what is this regulatory protein? This is a protein. So this mRNA molecule is now functional. This is a mature mRNA molecule. Pre-mRNA molecule was so the intron was removed from the pre-mRNA molecule. Exons were put together, and this is now a mature mRNA molecule. This mRNA molecule is now translated by the free ribosomes inside the cytoplasm, and this translation results into the formation of a gene regulatory protein. This gene regulatory protein then enters into the nucleus through the nuclear pore complex, and this binds to the regulatory sequence of a gene, and this is a gene for chaperones. So regulatory sequences are some untranscribed regions. So they're the regions which are not transcribed, but there are certain sequences which can be recognized by certain proteins, and these proteins would bind to them. Like you have your promoter sequence, enhancer sequence, operator sequence. So these are, all of them are regulatory sequences. So this protein basically would act as a transcription factor. And what is a transcription factor? A factor that can upregulate or downregulate the transcriptional activity is known as a transcription factor. So this is a gene regulatory protein. This would enter into the nucleus and would act as a transcription factor and would bind to the regulatory sequence of this gene. When it binds to the regulatory sequence of this gene, this is a gene for chaperones. So this gene is upregulated and starts synthesizing the mRNA molecules for chaperones. These mRNA molecules once again are exported out of the nucleus into the cytoplasm, but these are not synthesized by the free ribosomes inside the cytoplasm. Rather, this mRNA would again be recognized by the signal recognition particle and would be recruited onto the translocon in the ER. Now, once it is recruited to the translocon in the ER, the synthesis of the polypeptide chain the polypeptide chain is later pushed into the into the lumen of the ER via co-translational translocation. So this is being synthesized and being pushed into the lumen of the ER at the same time. And once this is inside the lumen of the ER, these are chaperone proteins. These chaperone proteins would bind to the misfolded proteins. And why do they bind to these misfolded proteins? So one of the reasons why most of the, not all, but most of the heat shock proteins are also chaperones. What chaperones do, they tend to block aggregates of the proteins, and this is very common when there's proteins tend to deform with temperature. So if you increase the temperature, there are pretty good chances that a rise in temperature would degrade your proteins. And when proteins are degraded, they tend to stick together, they tend to make clumps, they tend to make aggregates. So what the chaperones do, the binding of chaperones to these misfolded proteins, which have been misfolded for one reason or the other, binding of these chaperone proteins to these misfolded proteins basically prevents or inhibits these aggregations. And then what happens with these proteins which are bound or associated with the chaperones, they need to be degraded. And there's no such mechanism inside the ER to degrade these proteins. So once again, these proteins need to be exported out of the ER. So previously they were synthesized and inserted into the ER, but they were misfolded or later became misfolded. They triggered the synthesis of chaperones via activation of a transmembrane protein kinase. This kinase removes intron from a pre-mRNA molecule and converts it into a mature mRNA molecule. This mature mRNA molecule is then translated into gene regulatory protein. This gene regulatory protein enters into the nucleus and activates, binds to the regulatory sequence of the chaperone's mRNA. Chaperone mRNA is synthesized, comes back into the cytoplasm and is translated onto the ER membrane. And chaperone proteins are being synthesized directly into the lumen of the ER. But this protein, which is misfolded, it was previously synthesized via the same mechanism into the lumen. It now needs to be exported out. So two things are being done here. Two functions are being performed. One, chaperones are preventing these proteins from aggregations, from making clumps. And second thing, they will export them out of this ER back into the cytoplasm so that they can be degraded. And why do we need to degrade them? Because if they're not properly folded, they would not only accumulate inside the ER, inside the cytoplasm, but they would also cause hindrance to the normal functions of the cell. If they keep aggregating and they're non-functional, they're not being utilized, they would jam up the entire process inside the cell. So these proteins which became associated with the chaperones are now once again exported out of the ER lumen into the cytosol. And the interesting thing is this takes place through the same translocator, that is Sec61 complex. So this misfolded protein is exported out through this Sec61 translocator back into the cytoplasm. But these translocators contain an accessory exit protein unit associated with them, which helps in the export of this misfolded protein out into the cytoplasm. So once this protein reaches inside the cytoplasm, if this misfolded protein was associated with some sort of carbohydrate molecule, means it was a glycoprotein was associated with the carbohydrate molecule, we first need to remove this carbohydrate molecule from the protein. And this function is performed by an enzyme known as N-glycanase. So N-glycanase would remove the carbohydrate molecule from the protein. So that's deglycosylation. Previously it was glycosylation. The protein was glycosylated in the ER, but because this protein now needs to be degraded, it first needs to be deglycosylated, and that is this function is carried out by the enzyme known as N-glycanase. So N-glycanase would remove the carbohydrate molecule, and this protein would become deglycosylated. So it's not glycosylated anymore. Now this protein needs to be targeted to another machinery, small machinery known as proteasome. Now this proteasome is an organelle which is responsible for the degradation of the proteins. But proteins cannot on their own move towards the proteasomes, and they need to be targeted. And this targeting is carried out by another smaller protein which is known as ubiquitin. So this is a pretty small protein consisting of few amino acids. So multiple molecules of ubiquitin are associated with this protein, the protein that needs to be degraded. So binding of multiple ubiquitin molecules basically leads to, so this process is also known as conjugation. So this means conjugation or binding of the ubiquitin molecules to the protein that needs to be degraded. So binding of these ubiquitin molecules to this protein targets this protein towards the proteasome. So this protein goes into the proteasome, and proteasome has got lots of different types of enzymes which can carry out functions of deconjugation or deubiquitination. Means ubiquitin molecules would be removed from this protein inside the proteasome, and this protein would be then degraded into smaller pieces or into individual amino acids, and then these smaller chunks are then exported out back into the cytoplasm, and these amino acids would then be reutilized in the synthesis of some other proteins by the free ribosomes which are inside the cytoplasm. So this is how proteins are recycled. So the proteins which were not functional, which were misfolded, these proteins are brought back into the cytoplasm out of the ER with the help of chaperones. They're deglycosylated, and ubiquitination then targets these proteins into the proteasomes, where they are degraded and their individual entities that are amino acids, these are thrown back into the cytoplasm so that they can be utilized to synthesize some new proteins. I think that's sufficient for today. Thank you very much for your attention. So if you have got any questions, you can post these questions on Google Classroom, and I would be glad to answer all of your questions. Thank you very much for your attention.